Welcome to the MCC International Webinar Series. This is brought to you by the Mabalacat City College, Office of the Vice President for External Affairs, and the Institute of Arts, Sciences, and Teacher Education. For our first webinar episode on advanced biology, our speaker is Mr. Mark Tristan Kimke. He is a faculty member at the Department of Chemistry, Mindanao State University, Iligan Institute of Technology. Currently, he is also a PhD candidate for the degree of Doctor of Philosophy in Chemistry at the University of Santo Tomas. His topic for today is on computational approaches to COVID-19 drug discovery.
Good day. I am Mark Tristan Kimke, a faculty member of the Department of Chemistry of the Mindanao State University, Iligan Institute of Technology, and currently a candidate for the Doctor of Philosophy in Chemistry at the University of Santo Tomas. My research interests are chemical synthesis, natural products chemistry, and computational chemistry. Today, I will be presenting a talk which I think is timely and relevant to the current situation we are in, entitled Computational Approaches to COVID-19 Drug Discovery. Before I start, here's the outline of my talk, the 7 C's of my presentation, if you will. We'll start with a brief introduction of COVID-19 and the challenges in the drug discovery process, particularly for this disease, how computer-aided methodologies may help with these challenges, and how it fits into the drug discovery pipeline. I will also discuss some of the computational tools available to us. Then we'll talk about central dogma, specifically the life cycle of SARS-CoV-2, and lastly, I will share with you some of the highlights of our current paper on virtual screening-driven drug discovery of SARS-CoV-2 enzyme inhibitors. It's really a difficult time we are in. The word COVID-19 has been connotated with the words pandemic, with fear, with the new normal. Over the past few months, our lives, one way or another, have been affected by this disease. Some more drastic than others, but nonetheless, it was never how we imagined our year 2020 would be. This is the culprit, the novel coronavirus SARS-CoV-2, which is the causative agent of the disease COVID-19. Since December 2019, it has caused rapid transmission across countries, forcing lockdown of communities in many countries, affecting not only health, but caused a huge negative impact on the social and economic well-being of individuals and the world in general. This prompted the World Health Organization to declare COVID-19 as a global pandemic on March 11, 2020. There is no licensed drug or vaccine available for SARS-CoV-2, although several drugs have been repurposed and currently undergoing clinical trials to test their efficacy and safety in the treatment of COVID-19, such as the following: the antiviral drug favipiravir, under the trade name Avigan, used to treat influenza; chloroquine and hydroxychloroquine, which had been approved for the treatment of malaria, lupus, and rheumatoid arthritis; remdesivir, a nucleotide analog prodrug that inhibits viral RNA polymerases, which was tested against Ebola virus; the anti-HIV drug cocktail lopinavir and ritonavir; and an anti-inflammatory corticosteroid dexamethasone, to name a few. However, given the rippled effect of this pandemic and the lack of promising target-specific drug, an urgent call to discover effective therapeutic agents to combat the SARS-CoV-2 virus is still very much needed. While we need a safe and effective drug right now, it's always easier said than done. The discovery of a drug is a very complex process, which includes interdisciplinary efforts for designing effective, safe, and commercially feasible drugs. As a matter of fact, the process of discovery and development is not only very challenging, but also expensive and time-consuming. For a highly contagious epidemic such as COVID-19, the scientific community is cowering and pressured in search for a cure and a vaccine. And we all know that this scientific endeavor is a race against time. Over the past few years, modern drug discovery has been accelerated due to the development and advancement of bioinformatics and computational methodologies. Computer-aided drug design, or CAD, also known as in silico screening, has become a powerful technique because of its utility in various phases of drug discovery and development. In silico screening also paves path for the synthesis and screening of compounds for better therapeutics. Among the advantages of CAD are the following. It requires much less investment in technology, resources, and most importantly, the much dreaded factor in the case of COVID-19, time. The employment of CAD consequently reduced the expense, failure, and hastened the research cycle of drug discovery. Computational methodologies have now become a vital adjunct to the drug discovery process. We'll now discuss the computational or in silico drug discovery pipeline. As illustrated, the process occurs in 3 stages. The initial phase is target identification. Before you can develop a drug, there needs to be a specific target. This could be a protein, for example, which drives disease progression, by which you design your drug around. And for a successful computer-aided drug design research, access to an acceptable and validated 3-dimensional target protein structure is vital. This is where bioinformatics and structural biology comes in. In the case of COVID-19, the complete genome was made available last January this year, which can be retrieved from the National Center for Biotechnology Information with the following NCBI reference sequence number. Sites such as Swiss Model by the Swiss Institute of Bioinformatics of the University of Basel, Switzerland, Zhang Lab by the University of Michigan, and the RCSB Protein Data Bank, which provided a genome-wide structure and function modeling of SARS-CoV-2. We were able to take advantage of the available protein structures for our in silico studies. Remember, the choice of a drug target is primarily made on a biological and biochemical basis. After deciding on a particular protein as a drug target, computational methodologies can aid in lead discovery and optimization. The word lead refers to a chemical compound that shows promise as a treatment for disease and may pave a way for the development of a new drug. The commonly used in silico lead identification and optimization methods can be categorized into either as structure-based and ligand-based approaches. Structure-based drug design or SBDD methods, which includes molecular docking, molecular dynamic simulation, and de novo drug design, rely on the knowledge of the structure of the target macromolecule. Some of these methods will be discussed in a few. In the absence of the 3D structures of potential targets, ligand-based drug design or LBDD tools, which includes quantitative structure-activity relationship or QSAR, pharmacophore modeling, etc., can provide crucial insights into the nature of the interactions between drug targets and ligands, which allows predictive models that are suitable for lead discovery and optimization to be constructed. QSAR analysis in particular is a widely used technique in cheminformatics for predicting a response variable given a set of molecular descriptors such as structural, chemical, and/or physical input variables. This method may require experimental data such as in vitro or in vivo analysis for a successful prediction. But unfortunately for COVID-19, we do not have enough or even have access to these data yet. The third stage of the pipeline is the subjection of the lead compounds to preclinical tests such as drug likeness prediction studies. This involves predicting particular properties based on a drug candidate structure. Properties of interest include the following: structural properties such as hydrogen bonding, polar surface area, and lipophilicity, physicochemical properties such as solubility, permeability, and chemical stability, and pharmacokinetic or ADME and toxicity properties. ADME stands for absorption, distribution, metabolism, and excretion. Prediction of ADME by in silico tools has now become an important part in pharmaceutical research. Recently, machine learning as well as non-linear statistical tools have been widely applied for such applications. Allow me to give a brief discussion on some of the common computational tools available to us. For this purpose of the presentation, I will not delve any more in the math and the computational algorithm, so this will be more of a descriptive discussion. Molecular docking is one of the frequently used methods in SBDD because of its ability to predict with a substantial degree of accuracy the conformation of small molecules, which acts as ligands, within the appropriate target binding site of the protein, which acts as the receptor, to form a stable complex. Basically, it attempts to find the best match between molecules. When it comes to ligand-protein interaction, what commonly comes to mind is the problem on lock and key, meaning that a ligand acting as a key should have the right geometry to enable it to fit to the keyhole of the lock, which is the protein, and probably acts as an inhibitor to attenuate or minimize protein activity. However, since both the protein and the ligand structures are flexible, which is said to be so during program calculation, the lock and key model is now less accurate. During the course of the docking process, the ligand and the protein adjust their conformation to achieve an overall best fit, and this kind of conformational adjustment is referred to as induced fit. In here, investigations involving crucial molecular events are conveniently performed, which includes ligand binding modes and the corresponding intermolecular interactions that stabilize the ligand-receptor complex. Furthermore, molecular docking algorithms execute quantitative predictions of binding energetics, providing rankings of docked compounds based on the binding affinity or binding energy of respective complexes. Although proteins are normally represented as static structures, they are in fact dynamic. We have learned earlier that these macromolecules have degrees of flexibility in them, particularly when accommodating ligands during complex formation. In other words, a binding event requires flexibility of the protein. Dynamics in this context represents intrinsic subregional motion, which can be quantified and described in terms of kinetics. With these in consideration, molecular dynamics, or MD, is another principal tool in the theoretical study of biological molecules. It basically calculates the time-dependent behavior of a molecular system, meaning the ligand-protein complex, as it undergoes conformational changes during the molecular recognition process, where detailed information on deviations and fluctuations through time are noted. Moreover, MD is also used to investigate the structure, dynamics, and thermodynamics of biological molecules and their complexes. MD simulations are more accurate, but are often more computationally costly. As such, a hierarchical strategy is often employed, wherein a library of compounds is screened using molecular docking scoring function, which is efficient and high throughput, prior to subjecting a selection of best scoring ligands to MD simulations. In drug discovery and development programs, understanding the pathogenesis of the disease is very important. This has been stressed earlier, particularly on the basis of choosing the drug targets. For COVID-19, the life cycle of SARS-CoV-2 begins with the binding of its viral spike through host cell receptors of the cell membrane. The primary entry of the virus is mediated by ACE2 or angiotensin-converting enzyme 2 receptors, forming complexes with these proteins. Another possible route of entry to the host cell, a secondary mode of entry if you will, is through binding with the chaperone protein glucose-regulated protein 78, or GRP78. It was presented by the study of Ibrahim et al. That GRP78, while localized in the lumen of the endoplasmic reticulum, can be translocated to the cell membrane during cellular stress, possibly aiding entry of the virus. Now, after the virus enters the host cell through endocytosis, uncoating of the virus occurs in the host cell. The uncoated viral RNA is consequently used as template to directly translate polyproteins, whose function is to encode 16 non-structural proteins, or NSPs. This includes cysteine proteases, the papain-like protease PL-Pro, and the chymotrypsin-like protease 3CL-Pro. PL-Pro, a part of NSP3, functions to cleave the N-terminus of the polyprotein to release itself along with NSP1 and 2, while 3CL-Pro cleaves the remaining NSPs 4 to 16. Hence, cysteine proteases may be utilized as drug targets to inhibit post-translational modification and activation of viral enzymes. Other drug target-worthy enzymes include the RNA-directed RNA polymerase, or RDRP, which is important in viral replication, and NSP15, which prevents simultaneous activation of the host cell dsRNA sensors, thus inhibiting detection of the host immune system. And now, I would like to showcase the highlights of our study, published in the Journal of Biomolecular Structures and Dynamics, entitled Virtual Screening-Driven Drug Discovery of SARS-CoV-2 Enzyme Inhibitors Targeting Viral Attachment, Replication, Post-Translational Modification, and Host Immunity Evasion Infection Mechanisms. Our study focused on the exploitation of fungal secondary metabolites, particularly a library of 97 metabolites, with profound antiviral activity on a range of known pathogenic viruses such as the human immunodeficiency virus or HIV, influenza A H1N1 virus, herpes simplex virus or HSV, hepatitis C virus or HCV, as potential drug prototypes against the SARS-CoV-2 virus. Here are the 5 target proteins crucial for the infectivity of SARS-CoV-2 that were selected as molecular targets. The homology model of the catalytic domains of the target proteins was generated in the Swiss model workspace using high-resolution crystal structures. Coach algorithm was used to do a stochastic search for putative binding sites. Molecular docking experiments, which includes ligand and protein preparation, were performed on the UCSF Chimera platform, employing the BFGS algorithm of Otto Dacvina for the docking simulations. Ligand preparation was done using Avogadro software, and Biovia Discovery Studios was used for visualization of the complexes as well as post-doc analysis. Molecular dynamic simulation and relative free energy calculations was done on Amber 18 molecular dynamics package. Swiss ADME was used for the in silico ADME or pharmacokinetic prediction, and Osiris Property Explorer was used for the toxicity predictions. Let me now share with you some of our results, starting with the first target enzyme, PLpro. This is a multi-domain protein and is the largest replicase unit. It is responsible for the cleavages located at the N-terminus of the replicase polyprotein. Through in silico-based screening, 13 fungal secondary metabolites displaying high affinity towards PLpro were noted. Among these compounds, the fumiquinazoline marine alkaloids cetapin C and norquinadolin A demonstrated highest affinity to PLpro, both with binding energies of -10.9 kilocalories per mole. Another quinazoline alkaloid, quinadolin B, also showed high affinity with binding energy of -10.6 kilocalories per mole. Comparison of the docking behavior of the top 3 inhibitors for SARS-CoV-2 PLpro revealed that these alkaloids all contain a quinazolinone core primarily involved in interacting with PLpro's putative binding site. The second cysteine protease is 3CLpro, which functions by cleaving the polyprotein at 11 distinct sites to generate other non-structural proteins vital in viral replication. The 3CLpro monomer has 3 domains, wherein the active site, the one encircled, housing the catalytic dyad cysteine 145 and histidine 41, is located between domains 1 and 2. The most notable inhibitor is the anti-HSV metabolite from the marine-derived fungus Aspergillus tyreus, 11A-dehydroxyisoteriolactone A. An unbiased or blind docking was also performed for this compound, meaning we did not direct the docking to the perceived binding site in order to assess the preferential attachment of the ligand to the active site as opposed to other binding pockets. Interestingly, this compound adapts to the same docking mechanism within the binding site of the catalytic dyad. Alkaloids cetapin C and sequinadolin A also showed significant binding affinities towards 3Cl-Pro. These two compounds were isolated from the marine fungus Pseudosporium aspupermum and displayed significant antiviral activity against hepatitis C. RDRP is an essential protein encoded in all RNA-containing viruses such as SARS-CoV-2, responsible for catalyzing the synthesis of the RNA complementary strand, thus has a vital role in viral genome replication. The active site of RDRP is highly conserved and contains aspartate residues that protrude from the beta turn joining the beta 15 and beta 16 strands. The results of this study showed that the top 3 RDRP inhibitors are quinadolin B, cytokalasin Z8, and cetapin C, which were previously known to exhibit activities against RNA-based viruses, specifically HIV, influenza, and hepatitis C, respectively. NSP15 is an indoribonuclease that cleaves 3' of urilidates through ribonuclease A-like mechanism. It inhibits detection of the host immune system. It does this by preventing simultaneous activation of host cell dsRNA sensors. The structure of SARS-CoV-2 NSP15 is quite similar to other coronavirus NSP15 monomers, which consists of 3 distinct regions: the N-terminal domain, a subsequent middle domain, and a catalytic C-terminal nidoviral RNA urilidate-specific indoribonuclease domain. Generally, for coronaviruses, the C-terminal domain houses the active site of the enzyme, specifically the proposed catalytic triad histidine 235, histidine 250, and lysine 290. 3 secondary metabolites, namely isoketochromine D1, quinadolin B, and cetapin C, exhibited remarkable binding mechanisms to NSP15. Each were bound to the enzyme, hinging on NSP15's catalytic triad of the C-terminal domain through H bonding, pi interactions, and van der Waals stabilization. The spike protein is a homotrimer protruding from the outer surface of the virion and serves as the major driving force for host cell recognition and entry. Although the primary entry of the virus is mediated by ACE2 receptors, it was mentioned that the spike may act as binding sites for the GRP78. During cellular stress, this master chaperone protein, GRP78, is overexpressed, which increases the chance for it to be translocated from the ER to the cell membrane, making it susceptible to virus recognition. Here, ligand docking is directed to GRP78 binding region of the spike protein, particularly at region 4 of the spike, amino acids residue 480 to 488. Also shown on the screen are the top 3 binding ligands of spike protein. In this study, we used MD simulation to understand the dynamics behavior of our top scoring compounds in complex with their respective proteins. Post-simulation analysis for stability and root mean square deviation, or RMSD, of all ligand protein systems were calculated within a 50 nanosecond time frame. It can be seen that all systems in the bound state attain dynamic stability. To further understand the binding of the top scoring compounds, a total binding free energy was calculated using MMPBSA approach, and all tested ligands possess strong inhibitory effect against the specific target. The 25 high DOC scoring compounds were submitted for in silico ADME screening using Swiss ADME to predict their overall pharmacokinetic behavior. The drugability of each compound was assessed based on Lipinski's rule of 5, which is primarily focused on the prediction of the following descriptors: molecular weight less than 500 Daltons, lipophilicity reported as octanol water partition coefficient less than 5, and the number of hydrogen bond donors less than 5 and acceptors less than 10. Likewise, the boiled egg model, or brain or intestinal estimated permeation predictive model, is an intuitive graphical plot of the functions of lipophilicity described by the parameter W log P, or atomistic octanol water partition coefficient, and apparent polarity described as topological polar surface area, or TPSA. This is used to predict passive intestinal absorption and brain permeation of the compounds. Compounds located in the yellow region, or the yolk, have a high probability of blood-brain barrier penetration, while those in the white region have the propensity for passive absorption through gastrointestinal tract. A prediction of the toxicities of the top-ranked compounds was also performed using Osiris Property Explorer. Toxicity risks include mutagenicity, tumorigenicity, irritant effect, and reproductive toxicity. In here, we screen for top-dock compounds with low toxicity risks and with good oral bioavailability and druggability based from ADME predictions. To summarize, we were able to perform virtual screening of various fungal metabolites on the basis of molecular docking behavior against SARS-CoV-2 target proteins, which are dynamically stable and confer favorable ADME-tox properties. Thus, we disclosed the in silico aided discovery of two fumiquinazoline alkaloids, quinadolin B and cetapin C, and polyketide isoketochromine D1, exhibiting strong activity against not one, but 5 proteins involved in SARS-CoV-2 infection, such as viral attachment, replication, post-translational modification, and evasion of host immunity mechanisms. Therefore, these compounds can be utilized as templates or prototypes for further development of multi-target ligands against SARS-CoV-2. I would like to end my talk with this quote from Nobel laureate John Bardeen: "Science is a collaborative effort." With that said, I wish to acknowledge the team behind this research, comprised of chemists, a microbiologist, medical student researchers, a pharmacist, and computational biologists, headed by Dr. Alan Patrick Macabeo. To my research teammates from the University of Santo Tomas, Ken, Ray, Renz, Andrew, Justin, Luis, Jael, and Adriel, and our collaborators from Shanghai Jiao Tong University of China, Dr. Kan and Dr. Wei. Also, thank you to the organizers of the MCC International Webinar Series for the opportunity. Thank you everyone for listening, and keep safe.
On behalf of Mabalacat City College OIC President, Honorable Mayor Crisostomo C. Garbo, and the Chief Administrative Officer of MCC, Ms. Michelle Aguilar Ong, we would like to thank Mr. Mark Tristan Kimke for sharing his time, effort, and expertise as our resource speaker for today. And on the next episode of our MCC International Webinar Series, we will be having Ms. Kay-Ann Baritugo. For more information, you may visit our official Facebook page. Thank you.